Gene editing is a revolutionary technology that allows us to directly manipulate the genetic code, the very blueprint of life. This powerful tool has immense potential in the field of medicine, opening up new avenues for treating genetic disorders, fighting diseases, and even enhancing human capabilities. The image conveys the cutting-edge, futuristic nature of gene editing, with the vibrant, glowing DNA strand, symbolizing the transformative power of this technology. As we delve deeper into the science of gene editing, we must consider both the remarkable benefits and the ethical considerations that come with altering the fundamental building blocks of life. Gene editing is the process of precisely altering the genetic code, allowing us to modify, insert, or delete DNA sequences. This technology enables us to target specific genes and make changes to produce desired traits or characteristics. Gene editing provides a powerful tool for understanding and manipulating the fundamental building blocks of life. The ability to edit genetic information has significant implications for fields like medicine, agriculture, and scientific research. The history of gene editing is a fascinating journey, spanning over a century of scientific progress. In the early 20th century, researchers began to uncover the fundamental building blocks of life, genes. In the 1970s, the first gene editing techniques were developed, opening up new possibilities for genetic manipulation. During the 1990s, early gene therapy trials were conducted, exploring the potential to treat genetic disorders. The 2000s saw the development of zinc finger nucleases, a powerful tool for targeted gene editing. And in the 2010s, the CRISPR-Cas9 system revolutionized the field, making gene editing more accessible and precise than ever before. This timeline highlights the remarkable advancements that have led us to the cutting edge of genetic engineering today. Gene editing technologies allow us to precisely modify DNA sequences. I'll briefly cover 4 key gene editing tools: restriction enzymes, zinc finger nucleases, TALENs, and CRISPR-Cas9. Restriction enzymes are molecular scissors that cut DNA at specific sites. Zinc finger nucleases are programmable DNA-binding proteins that can be designed to target desired sequences. TALENs are also customized DNA-binding proteins, similar to zinc finger nucleases. CRISPR-Cas9 is an RNA-guided DNA cutting enzyme that is highly versatile and efficient for gene editing. CRISPR-Cas9 is a revolutionary gene editing tool that has transformed the field of genetics and biotechnology. It is derived from a natural defense mechanism used by bacteria to protect themselves from viral infections. The system consists of 2 key components: the Cas9 enzyme, which acts as the molecular scissors to cut DNA, and the guide RNA, which directs the Cas9 enzyme to the specific DNA sequence that needs to be edited. This powerful combination allows for precise, efficient, and targeted modifications of the genome, opening up countless possibilities for research, medicine, and beyond. CRISPR-Cas9 is a powerful gene editing tool that offers several key advantages. First, it is highly precise, allowing us to target specific DNA sequences with great accuracy. This targeted approach is a major advantage over previous gene editing methods. Second, CRISPR-Cas9 is remarkably efficient, with a high success rate in successfully editing genes. This efficiency makes it a reliable and effective tool. Third, CRISPR-Cas9 is versatile. It can be applied to a wide range of organisms and used to address various genetic diseases and disorders. Its broad applicability is a significant strength. Finally, CRISPR-Cas9 is more cost-effective compared to other gene editing technologies. This makes it more accessible and opens up new possibilities for research and therapeutic applications. Off-target effects are a key limitation of CRISPR-Cas9. The system can cut DNA at unintended sites, leading to undesirable genetic changes. Delivering the CRISPR-Cas9 components efficiently to the target cells is an ongoing challenge. This is critical for the technology to work as intended. Mosaicism is another issue. Not all cells in an organism may be successfully edited, leading to inconsistent and unpredictable results. The Cas9 protein itself can trigger an immune response in some cases, which is an important consideration for therapeutic applications. Overall, while CRISPR-Cas9 is a powerful tool, these limitations must be carefully addressed as the technology continues to develop. Gene editing has a wide range of applications, from agriculture to medicine. In agriculture and food production, we can use gene editing to enhance crop yields, improve resistance to pests and diseases, and boost the nutritional content of foods while reducing waste. In the medical field, gene editing holds immense potential for treating genetic diseases and disorders. It can enable the development of new therapies and diagnostic tools. The versatility of gene editing technology is truly remarkable, and we're just beginning to scratch the surface of what it can achieve in transforming our world. Gene editing has enabled us to develop crops with improved resistance to pests and diseases, leading to higher yields. We can now modify crops to be more nutritious, boosting their vitamin and mineral content. Drought-resistant crops created through gene editing can better withstand water scarcity, improving food security. Genetically modified crops that are naturally resistant to pests can reduce the need for harmful pesticides. Medicine and healthcare are advancing rapidly, driven by new discoveries in genetics and personalized treatments. One key area is treating genetic diseases by correcting the underlying genetic defects that cause them. Another important trend is personalized medicine, tailoring treatments to the unique needs and genetic profile of each individual patient. We are also seeing the development of novel gene-based therapies, which offer new ways to treat diseases. And in cancer treatment, we are getting better at targeting cancer cells with precision, improving outcomes for patients. These advancements are made possible by our growing understanding of the human genome and how to harness it for better health. Environmental conservation is crucial for addressing climate change and protecting our planet's natural ecosystems. Reforestation is an effective way to reduce carbon emissions and support biodiversity by planting more trees. Water conservation, through efficient irrigation and reducing waste, is essential, especially in regions facing water scarcity. Pollution control, including reducing emissions and waste, helps safeguard ecosystems and the health of our communities. Preserving habitats and protecting biodiversity is key to maintaining the rich variety of species that are vital to the balance of our natural world. The vibrant coral reef seen on this slide represents the beauty and diversity of our marine environments, which we must work to conserve. The topic of this slide is the ethical considerations around gene editing, specifically informed consent and genetic privacy. When it comes to informed consent, it's crucial that we fully inform individuals about the risks and benefits of gene editing and ensure they have the capacity to understand the information and make autonomous decisions. On the topic of genetic privacy, we need to protect sensitive genetic information from unauthorized access and misuse, and ensure individuals have control over their genetic data. These are two key ethical issues we need to carefully consider as we develop and apply gene editing technologies. The global network of researchers, scientists, and ethicists shown in the image represents the collaborative nature of this field and the need for international cooperation. Harmonizing regulations for scientific research and clinical trials is crucial to ensure consistency and fairness across borders. Ethical oversight is essential to guarantee the responsible and beneficial use of this technology, protecting individual rights and societal well-being. Public engagement and education are key to building trust and understanding around the potential of this technology and its associated risks. Rigorous risk assessment is necessary to identify and manage any unintended consequences or negative impacts that may arise from the development and deployment of this technology. Gene editing tools must undergo rigorous testing to ensure they are safe and effective before being used. Ethical oversight is critical to address the potential risks and unintended consequences that could arise from gene editing. Minimizing off-target effects, or unintended edits to non-target genes, is a key priority when working with gene editing technologies. Careful consideration of these safety and risk management factors is essential when utilizing gene editing tools to avoid potential issues. The image on this slide depicts the key difference between germline and somatic cell editing. Germline changes are inherited by offspring, while somatic cell changes only affect the targeted cells. Germline editing has the potential to pass on genetic modifications to future generations, which raises significant ethical concerns that we'll need to carefully consider. In contrast, somatic cell editing only affects the individual being treated, without altering the germline. This can be a useful approach for treating certain genetic diseases. As we discuss the pros and cons of each approach, it's important to weigh the potential benefits against the ethical implications to ensure we are making responsible decisions. Off-target effects are a key challenge with CRISPR technology. CRISPR can sometimes cut DNA at unintended locations, leading to harmful mutations. This is an important issue that needs to be addressed. Researchers are working on improving CRISPR designs to minimize off-target effects. Rigorous testing and analysis is critical to identify and mitigate off-target edits. Careful validation is required before using CRISPR in clinical applications. Mosaicism refers to the presence of genetically distinct cell populations within an individual, often due to genetic changes that occur after fertilization. Some cells in the body may have an edit or change, while others do not. This can lead to a mosaic pattern. When the edit is incomplete or partial, it can result in unpredictable outcomes, as different cells have different genetic makeups. The complex effects of mosaicism may impact the effectiveness of certain treatments, as the genetic diversity within the individual can influence how they respond. It's important to understand the implications of mosaicism, as it can have significant implications for diagnosis, prognosis, and treatment planning. The immune system is our body's defense mechanism, designed to detect and eliminate foreign invaders, like viruses and bacteria. However, gene editing can sometimes trigger an unintended immune response, where the body views the edited cells as foreign and attacks them. This immune system attack can lead to inflammation and other complications that need to be carefully managed. We need to be mindful of the potential for the immune system to react adversely to gene editing and take steps to mitigate these risks. Gene editing tools can be delivered using modified viruses as vectors. Lipid nanoparticles and other non-viral methods are also options for gene delivery. Direct injection of the gene editing tools into target cells is another approach. Each delivery method has its own advantages and considerations in terms of efficiency, safety, and targeting. The choice of delivery method will depend on the specific application and requirements of the gene editing therapy. Viral vectors are modified viruses that can be used to deliver genetic material into cells. They have the advantage of being an efficient way to get genes into target cells. However, They can also trigger an immune response, which is a potential disadvantage. The two main types of viral vectors are adenoviruses and lentiviruses. Adenoviruses and lentiviruses have been engineered to remove their disease-causing properties while retaining their ability to deliver genetic payloads. Non-viral gene delivery methods are an important alternative to viral vectors. Nanoparticles can directly deliver genes into target cells. Lipid nanoparticles provide a safe and efficient way to deliver genetic material. Cell-penetrating peptides help facilitate the delivery of genes into cells. Electroporation uses electric pulses to temporarily open cell membranes, allowing genes to enter. These non-viral approaches offer advantages like improved safety and customizability. Sickle cell disease. This was the first successful gene editing clinical trial, showing the potential of this approach. Duchenne muscular dystrophy. Ongoing trials are showing promising results for treating this devastating disease. Cystic fibrosis. Trials are underway with positive outcomes. So far, these examples demonstrate the broad applicability of gene editing to treat a wide range of genetic conditions. Gene editing holds great promise to transform how we approach many devastating diseases. Sickle cell disease is a genetic disorder that causes the red blood cells to be sickle or crescent-shaped rather than the normal round shape. This abnormal shape can cause the cells to get stuck in small blood vessels, leading to painful episodes called vaso-occlusive crises. Sickle cell disease can also cause respiratory issues like difficulty breathing and shortness of breath. Treatment options include blood transfusions to help improve blood flow, as well as pain management strategies to deal with the painful crises. Duchenne muscular dystrophy is an inherited condition that affects muscle strength and movement. It's caused by a genetic mutation that leads to a lack of dystrophin, a key protein for muscle function. Symptoms typically appear in early childhood, with progressive muscle weakness. Children with Duchenne often lose the ability to walk by adolescence, as the condition continues to worsen over time. Cystic fibrosis is a genetic disorder that primarily affects the lungs, pancreas, and digestive system. The key issue is a buildup of thick, sticky mucus that can cause breathing problems and lead to infections. Researchers are exploring the potential of gene editing, specifically using CRISPR-Cas9 technology, to correct the faulty gene responsible for cystic fibrosis. Clinical trials are underway to investigate the safety and efficacy of this gene editing approach for treating cystic fibrosis. Gene editing technology has advanced rapidly in recent years, allowing for more precise and efficient tools. These advancements open up the possibility of personalized medicine, where treatments can be tailored to an individual's unique genetic profile. However, the societal impact of gene editing will require careful ethical consideration as this technology becomes more widely adopted. Key areas of concern include equity of access, unintended consequences, and the potential for misuse. As we explore the frontiers of gene editing, it will be crucial to have open and thoughtful dialogue about the implications for individuals and society. The rapid advancements in technology are transforming the future of healthcare, with key innovations in gene editing. Precision gene editing is becoming more accurate, allowing us to make targeted changes to genetic material with greater control. Delivery systems for gene editing tools are becoming more efficient, making the process easier and more accessible. We can now target specific genes with greater accuracy, enabling more personalized and effective treatments. The cost of gene editing is also decreasing, making these cutting-edge therapies more affordable and available to a wider population. These technological breakthroughs have the potential to revolutionize how we approach and manage a wide range of health conditions in the years to come. Personalized medicine is an approach that focuses on tailoring treatments to the individual needs and genetic profile of each patient. By leveraging genetic data, we can optimize treatments and therapies to be more effective and have fewer side effects for each patient. This leads to better health outcomes, as we are able to provide treatments that are precisely targeted to the individual. The key benefits of personalized medicine include tailored treatments, the use of genetic data to guide treatment decisions, and ultimately, improved effectiveness and reduced side effects. The potential societal impact of gene editing technology is a complex and multifaceted topic that we'll explore in depth today. One key consideration is the ethical debate around balancing the benefits of progress with the risks and challenges this technology presents. Another important aspect is the issue of social equality, ensuring equitable access to gene editing technology so that its advantages are not limited to only the privileged few. We'll also examine the economic implications, from the emergence of new industries to the potential impact on healthcare costs. Finally, we'll discuss public perception, how fears, hopes, and the need for greater understanding will shape the societal response to gene editing. These are all crucial factors to consider as we navigate the profound implications of this transformative technology. The potential of this technology is truly transformative. It could revolutionize fields like medicine, agriculture, and environmental conservation. However, with great power comes great responsibility. We must carefully consider the societal impact, ensure proper regulation, and prioritize safety as this technology develops. These two key points, the transformative potential and the ethical responsibility, are the core takeaways I want the audience to remember from this presentation. In summary, this technology holds immense promise, but we must approach it thoughtfully and with the utmost care for its implications.